Hey guys, in this video we begin the next unit of our ecology course, which focuses largely on populations. In terms of the ecological hierarchy, so far in this course we have looked at the bottom and at the top. So at individuals, what are the different characteristics, what are the different physiological features and adaptations that give rise to success in a particular environment. We've also focused all the way on the top of the ecological hierarchy, talking about the abiotic factors of the environment. We talked about climate in a lot of different ways and have kind of connected the individual all the way up to the environment. And so in this next unit, we are going to look at multiple individuals of the same species, that is a population, and how the populations grow or decline or soon interact with other species or populations of other species. And so this is information from chapter 8. This is of all of the units in this course, the most math heavy. In this lesson, I will show you one equation that we're going to use, and in the next lesson, we'll look at a lot more. So let's begin. So populations have unique features because they are aggregates of individuals, groups of individuals of the same species. And so it might seem kind of strange, but I ask you, what is an individual? Right, so we can think of ourselves, of course, as an individual. We grow from a single fertilized egg, so one genetically unique cell, and I say genetically unique because we are different genetically from both parents, right, from mom, from dad, and from that single genetically unique cell, those cells replicate into larger and larger forms. All of these cells work together. They are all roughly genetically identical, and they function together to form this whole discrete unit, that is the individual, that is us. But not every organism in the world is quite as easy to identify as an individual as something like a human would be. And so colonial animals are one example where it's a little bit difficult to define individual versus colony. So individual versus a population of interacting individuals. So in this Terebranch colony, these guys are actually living off of the coast of Antarctica. So they are maybe a little bit difficult to classify as an individual or not, because these organisms reproduce via budding. So one genetically unique individual forms a new bud, and that new bud becomes kind of an individual, but also still kind of connected back to the original animal. And so where does the individual stop and the colony begin? Right? So a little bit difficult. And when we're talking about plants, this gets even dicier. So you're probably familiar with sexual plant reproduction. Right, so here we have the stamens, they produce pollen. Pollen ultimately pollinates or fertilizes eggs, right, within the ovary of the flower down here. And once that fertilization event takes place, then a new genetically unique organism, right, forms as a seed or some other type of new structure. And so again, these are genetically unique individuals that are formed via sexual reproduction in plants, just like in humans, just like in animals. So we can call these genetically unique individuals gametes, but plants, as they grow more unique or discrete units, and these are different in different organisms and different even within the same organism. And so the organism grows a series of modules, right? So branches and leaves depending on environmental conditions, less so based on genetics. And so we can still look at this tree and say, okay, that is one individual, even though, you know, it has so many different discrete units as part of it. But during the process of growth, there are mutations, right? So now this individual, as it is growing, develops genetic mutations, and this discrete unit, this branch, and its associated leaves or needles is now genetically different from the rest of the tree, right? So is this a new individual or not? Is it just part of the same organism? And so sometimes this can be pretty fantastic. For example, a Sitka spruce in the Pacific Northwest was discovered to have like 100,000 mutations in this tree that was a couple hundred years old. And so, you know, if we are defining an individual as a genetically unique unit, is that Sitka spruce 100,000 different individuals, or, you know, what is it? And so we might say that single stem, and so the tree grows up out of the ground, that single stem defines an individual, even though different units on that individual are genetically unique. Right? So we could say that, but again, this is a little bit complicated. Some plants have multiple stems coming from the same root system. For example, grasses, multiple stems, same root system. Oak trees, another example, if you cut down an oak tree and you don't remove the stump, individual stems, in fact, many individual stems grow forth from that original root system. And so are they individuals, right? They're genetically the same. It's the same root system, the same genetics, but now we have multiple stems. So we can't really say one stem equals one individual, or, you know, one genetically unique unit is an individual, right? It gets a little bit complicated. Also, plants don't just reproduce sexually. They can also reproduce asexually in a variety of ways. So for example, here we see stolons, which are these lateral branches. In this strawberry plant here, they grow out and they can actually root down. And this, if you cut the stolon, can be its own independent plant, right? Genetically the same as the parent plant. All right, so this and this have the same genetics, but this is now its own unique unit, right? Own independent unit. And so this type of asexual reproduction happens above the ground here. It can also happen below the ground. Here we can see bamboo. This is a lateral root, and as the root expands out to all sides, it can send new shoots up, so new stems. Again, same genetics, but now we have multiple stems. Okay? And so these are ultimately clones, but is it a new individual? Or is it not a new individual? And so this isn't even just plants. We can also talk about other organisms, you know, fungi and sponges. Anything with asexual reproduction can produce this type of challenge to defining an individual. Just like a genetically unique individual is called a genet, a ramete is called is the term for these units produced by asexual reproduction. Okay, so we do have terms for both, kind of both definitions of an individual. Kind of the most extreme example of so many different rametes in one individual, one genetically unique entity, is the Pando clone, which is in Utah. If you consider one root system, one genome to be an individual, this is in fact the largest, most dense organism ever found in human history. And so it is nearly 13 million pounds. It covers 106 acres. So here we can see a forest that looks like all sorts of individual trees, these aspens here, but each one is a shoot off of the same root system. And so to look at this individual, we can't just like walk out into the forest and look up at a tree and say, there's the individual. In order to see the whole thing at the same time, we need to look from above, right? So this yellow line here is showing us the outline of this single clone, right? Maybe defined as a single organism, 106 acres, over 40,000 stems, right, rametes, in this single unique organism. And so it's thought that Pando actually grew from a seed at the end of the last ice age, and it just keeps on sending up new shoots and is seemingly immortal. Right? So really, really cool stuff, discovered in the 70s to be one single organism. And so just to add some terms here, this individual stems are the remnants of a single genetic organism, the genet. And so, again, it is challenging, of course, to define individual when we're looking at a population, like, is this a population of trees or is it an individual? And so generally, when these types of challenges are encountered by ecologists, the scientist has to define at what level they are studying, and studies happen at both levels. So just individual remnants, individual stems, as well as pando as a whole. Okay, so once we have defined what an individual is, some of the things that we can say about populations are, for example, the distribution. Okay, so the distribution is essentially determining the area where these individuals are versus where they're not, right? What is the distribution? We can map the position of each individual, okay? So each individual organism, and then draw a line around all of those individuals, all of those dots, and we can call that the distribution. And so this is just a hypothetical example. Each one of these red dots is an individual tree, for example. And so if we find every single one of the trees in this species in this area, we put them on a map, and then we draw a line around that map. That is the distribution. Okay. Similarly, we can kind of zoom out from here, zoom out from looking at the distribution and find all of the individuals of a particular species and, put a tracker on them, or, if it's a tree, something that stays in place. We can put a dot of where that animal or where that organism is on the map, and again, draw a line around it, and we can call that the geographic range. And so this is the geographic range of koala bears. Right, so everyone that we know about has been identified and put on the map here, and if we drew a line around it, we'd say that is the geographic range. Okay, organisms that move around a little bit more might require a different type of metric, a different type of technology, and so this is a distribution of polar bears. A GPS tracker has been put on a series of polar bears, and we can see their patterns of movement throughout the Hudson Bay. And so here, instead of just a dot, we actually have kind of the range of an individual animal. And so once again, we can just draw a line around all of these, and that would be the distribution of the polar bears. So the distribution of an organism is largely limited by that organism's zone of tolerance. Right? So connecting back to what we talked about on the very first day of class together. So animals, plants, fungi, all organisms can only live where the conditions are favorable for it. Right? They're going to be most abundant in places right in the middle of their zone of tolerance, and they kind of become less abundant towards the tails. In places where it gets colder or hotter, or some other type of environmental characteristic. So this here is the red maple distribution. So a type of tree turns beautiful red color in the fall, also produces lovely maple syrup if you harvest it in the right way. And so we can see that this tree, right, this tree's distribution, which is not everywhere, it's only this darker green, it has some limits, right? And the limits are due to its zone of tolerance. And so the upper limit is largely determined by the minimum winter temperature in Canada, right? So it can only survive through the winter at a certain temperature. If it gets any colder than that, as it would up here, farther in northern Canada, then the red maple would not survive in those places. And so other limits, of course, to the east and to the south, we have the ocean, right? It can't actually live in the ocean. That is well above its water and salt tolerance. And on the other side here in the Midwest, its limit is essentially due to the drier conditions within the Midwest, right? So along this east-west axis here, we can see the upper limits of water tolerance, right, and the lower limits of water tolerance. And so essentially we can imagine a zone of tolerance here. The optimal conditions are in this range. And so again, the zone of tolerance for different environmental characteristics determines the zone of tolerance. Right, another factor that limits the distribution of an organism are different types of barriers. Okay, so for example, maybe the weather is okay out here in the Atlantic Ocean, but again, the conditions are not okay. So this barrier or this ocean essentially acts as a barrier to this red maple spreading into Europe or into Africa. Okay, so even if the conditions were favorable for that plant, the ocean is serving as a barrier. Right, another example here, the Pine Barrens tree frog, which is endangered. It is in the New Jersey Pine Barrens, so right where Stockton is, hooray. This is the distribution of the Pine Barrens tree frog. We can see right down here in South Jersey, but we can also see a couple other patches of these organisms. And so the conditions that are favorable for Pine Barrens tree frogs are essentially evergreen oak forests. They like shrub bogs and really acidic water, acidic soil where they can lay their eggs. They are pretty tiny, and so it's difficult for them to cross barriers like roads, right? Like housing developments, like a lot of different anthropogenic barriers that are put into place. And so even though there are favorable conditions possibly throughout this range here, we don't see Pine Barrens tree frogs because it is difficult for them to actually cross these roads, cross these other barriers, and fully populate areas that are appropriate for them. Okay. And so again, lots of barriers keeping these populations small and isolated. Okay. Based on the distribution of an organism, we can describe them as being either ubiquitous or endemic. Or, it's a spectrum, something in between. So ubiquitous is essentially having a really wide geographical distribution. So the red maple, we could say, is ubiquitous throughout the eastern United States or eastern North America. Endemic, on the other hand, is a species with a relatively restricted distribution, native restricted distribution. So the Pine Barrens tree frog, very specialized habitat requirements and limited to very small distributions, limited to very small distributions. Even in the habitat range, the distribution of individual organisms, sometimes these species are not uniformly distributed. That is, even though we see that all of South Jersey is covered by Pine Barrens tree frog distribution, this dark green, in reality, you don't see Pine Barrens tree frogs just like on the lawn. You don't see them just around everywhere in parking lots or whatever. They have very specific habitat requirements. And so we can describe distribution at different spatial scales, from global down to local. And so here we see a global map, right? Zooming into just North America, we can see a few red dots throughout Canada and the United States, indicating where this particular species of moss is located. However, even though you see New Jersey pretty much covered by these red dots, that doesn't mean you're going to find this moss everywhere. So we zoom in even farther to South Jersey and along the Mullica River here, and we see these red dots are not covering the entire thing. They are just found in different places along the river. Right? So again, zooming in even farther, we see the river or the stream, and this moss is not found in the uplands and the forests. It's not found in the water, but instead it is found along the water, specifically along like swampy places. Again, zooming in even farther, we see that they are only found around the borders of coniferous stumps. So coniferous plants with the needles, only found right here. Okay, so within these really specific suitable habitats, you might see a ton of individuals, a ton of here's a plant and here's a plant and here's a plant. So they're all grouped together here. And so this species, right, we can talk about the distribution. Yes, it is, here we could draw a line around it, but in reality, within that distribution, they are not distributed equally. In fact, they are clumped. Metapopulations. Metapopulations are a collection of local subpopulation in patches of suitable habitat. So these guys are spatially separated, but they are still somewhat connected by migration or by genetic material moving back and forth, so reproduction across these different patches. So this is the distribution of mountain sheep. Right, so again, we can draw a line around all of it and say, yep, that is the distribution of this species. But in reality, this green space is not suitable for mountain sheep. This green space is not suitable for mountain sheep. And so we see that there are much smaller patches in which there are somewhat isolated populations, somewhat isolated groups of individuals. Okay? But we still see that there is some transfer of individuals and genetics between them. Okay. A good example of metapopulations we can see in agricultural fields. This generic agricultural area, we can see that between these patches of land with, of course, different species, first of all, there are these little hedgerows. So hedgerows essentially blocking the wind from blowing quite so hard and essentially reducing erosion of soil due to the wind. And so what we can see here is, an individual that lives in these shrubs, right? Their population, right? This population is somewhat isolated from this population because there's all these crazy fields in between. But there might still be some migration of individuals between these two metapopulations. Also, we could talk about the agricultural plants and the things that live on them as well. So maybe this is a field of grain and this is a field of grain. They are isolated for the most part, but because these species reproduce sexually via wind dispersal of the pollen, essentially this and these two metapopulations may still have gene flow between the two. Okay. A study of a pathogenic fungus on potato in China revealed just how much gene flow there was between metapopulations in different parts of the country. And so what we see here is the map of China. We have isolated populations, relatively isolated populations of this fungus in this part of China here in blue, another part in green, another part in blue and pink. And between some of these different populations, there is a considerable barrier to gene flow. Right, so you can think about the very high dense population of humans and other things in between, and just how much would it take for genes to flow from this potato field to the green potato field. But in their study, what these scientists found was that between the brown and the green, even though there are a lot of barriers between them, there's still considerable gene flow, right? Between brown and blue, right? Even bigger distance, so many more barriers, but still we see gene flow between the two. There is still connection between these subpopulations of the potato fungus. Right? Generally, what they found as I'm sure you could guess, is that humans really are the culprit. So you have machines going back and forth between these two places. We have trucks carting potatoes from here up to here, etc. And so this is how those metapopulations are being connected, is by human activity. Okay. Another thing that we can describe, or that we can use to describe populations, is abundance, right? So essentially, how many organisms are there within their distribution? And so something called population density, or sometimes crude population density, is used to describe how many organisms per unit of area. Okay, so essentially, again, all of these red dots are individual organisms. We have drawn a blue line indicating this is the distribution of random organism. Okay, if we place a grid over that distribution and sample a few, right, not just one, but a few of these cells of the grid, we can calculate crude population density. Okay, if we sample cell number 1, we can count 1, 2, 3, 4, 5 individuals. Okay, so if we only counted this one cell, this one quadrat, this one transect, whatever, we would say, yep, the density is 5 per meter squared. Okay, however, only sampling 1 is not going to give you very accurate results, and so generally we sample in multiple places. And so we'll sample, randomly another cell, and here we count 2 individuals, right? Sample again, we count no individuals within the cell. And so we have a density of 5, 2, or 0, depending on where in this distribution we sample. And so generally, this is what we see, or what we see in nature and in the human population as well. So if we looked at the human distribution, right, which is shown here in red, anywhere that has a slightly reddish tint, that is somewhere that humans live, so we pretty much live everywhere. Of course, the darker red indicates higher population, lighter red indicates lower population, right? Makes sense. But if we were to sample in Singapore, right, so over here, which is a lot darker red, we would count 8,000 people per square kilometer. On the other hand, if we were to sample in Greenland, we would count 0.2 people per kilometer, right? So very different densities depending on where we're actually looking. And so again, we should take the average of multiple places, right, multiple cells within the grid, in order to express population density. And so the average of this sample population here is 2.3 individuals per meter square. And sometimes you'll see more, sometimes you'll see less, but here's the average. And for the record, for humans, it's about 25 people per kilometer square, right? When you consider the entire human population, although these numbers are from a couple years ago, and so the number is really higher by now. Okay. The spatial pattern within the distribution can vary widely. Right? So essentially, just like we looked at before, even if you draw a line around where all the organisms are, depending on where you sample, there may be a lot of organisms or there may be very few organisms. And so ultimately, we can talk about spatial pattern using 3 different terms. There are 3 different patterns. Random distribution. So how are these organisms distributed within their larger distribution? We can say randomly. Okay, so here, what we see is a field of wildflowers. If we're looking at just the bluish purple flowers here, we see that they, there's a patch over here, and then they kind of fizzle out, and some over here. And so generally, these are called randomly distributed. There's really no pattern to it. There's not clumps, right? It's not evenly distributed, but they're randomly distributed. And so generally, this type of distribution is seen in organisms that have wind dispersal. So these plants, when they have, when they have seeds, the seeds are blown away by the wind, and just wherever they fall, that's where they're going to grow. So literally randomly. Uniform distribution. Again, this is where the organisms are. This is the distribution, but what is the pattern within that distribution? We can say it is uniform, right? So approximately equally spaced apart, such as we see here in these birds. Each one has its own tiny little territory for its nest, and all of them are kind of maximizing the space that's available to them in this particular space, in this particular distribution. Okay, we see this pattern a lot in animals that, or animals and plants that kind of have a territory, right? So multiple bears, right, like carving out their own piece of land, their own territory. They're going to expand as much as possible, and usually the territories turn out to be approximately the same size. Therefore, the animals themselves are approximately the same distance apart. Also, plants that compete for resources, again, carving out their own piece of land, they need so many resources, and so all of the plants maximize the space, and they all end up about equally spaced. Clumped distribution is by far the most common among all organisms on Earth. Here we can see, some space has no organisms and other spaces are very densely populated. Okay, so little clumps of these, in this case, fish. The school of fish, instead of spreading out individually, they clump together into schools for protection, of course. And so usually this is seen, organisms will clump together in the most suitable habitat, right? And that suitable habitat is not equally distributed. It is kind of patchy, right? This spot is great, that spot is not, and so everybody clumps together. Okay? Again, species from social groups like schools, like herds and flocks. And of course, if the organism reproduces asexually via budding, or stolons or whatever, the ramets themselves, right, are kind of clumped together. Okay. And so we can think about people for the most part as having a clumped distribution, right? So we form cliques in high school and even beyond, right? Or groups of friends kind of together. We clump together into cities for the most part, right? Until, of course, COVID happened, and then we artificially imposed this uniform distribution, right? Therefore, maximizing the space in between us, to protect each of us, of course. Okay, we have seen uniform distribution on day one of our class together. We talked about these termite mounds, right? So this uniform distribution is kind of implying that each one of these mounds is maximizing the space for protection, etc., around it. And last example here, we see in this forest, the orange colored trees, right? Sometimes there's more of them, sometimes there's less of them. It's kind of randomly distributed. Same thing for these conifers, which are still green, right? Kind of randomly dispersed. And this makes sense because these guys, wherever their seeds fall, that is where they're going to grow. Okay, so again, we can talk about the spatial pattern within the larger distribution of the organism. And that does, of course, tell us a lot about the organism. Now, sometimes determining whether something is uniform or clumped or random can be a little bit challenging. And so we have some math to help us decide or determine which type of spatial pattern is exhibited. And so in order to do so, we use what is called the index of dispersion, which is essentially the variance of all of the samples that we have taken. So all of the cells within the grid that we have counted over the average of those samples, of those cells. Okay. Variance here defined by this, and we'll take a look at this on the next slide, exactly how to go through. Of course, you can use Excel to calculate the variance. You can use Excel to calculate the entire index of dispersion. I'm going to show you I'm going to show you how to do this by hand. Okay, so the index of dispersion, the cutoffs here, if this value, this numerical value is less than 1, we call the population uniformly distributed. If it is approximately 1, we call it random. And if it's greater than 1, we call it clumped. Okay, so let's take a look. Once again, ecologists can learn a lot about the interactions between individuals within a population based on the distribution. Okay, so, for example, the trees competing for light may be uniform. Patchiness or heterogeneity might indicate that some parts of the habitat is great and some parts are less than great, and so organisms will clump together in only the best places. So now let's walk through this example that we already started before. Let's calculate the index of dispersion to figure out, are these actual clumps or is this more randomly distributed? Right? Are they uniformly distributed approximately? Like, what is the spatial pattern of this hypothetical species? All right, so again, the index of dispersion is the variance of all 3 samples that we've taken over the average of all 3 samples that we've taken. Okay, again, you can do this on the computer, but I'm going to show you how to break this down by hand. Okay, so the variance is the sum of all of the samples that you've taken. Right, so we have 3 different samples. The number of individuals in the cell, for example, 5, minus the average number of individuals among all of the cells sampled. That value is squared. Okay, so you do this calculation for 1, 2, and 3 cells, and then add all of those together and divide it by the number of cells sampled, so 3 minus 1. Okay, so let's walk through this. Previously, we determined cell 1 had 5, cell 2 had 2, and cell 3 had 0 individuals. Okay, the average of this we also determined to be 2.3, again from a previous slide. All right, so now we have the average, right? And we have this value, the number of individual cells in each cell. Or sorry, the number of individuals in each cell sample. Okay, so let's calculate this for each of the 3 cells. Okay, so 5 minus 2.3, the average, is 2.7. Squared is 7.29. Right? Again, 2 individuals in cell 2. So 2 individuals minus the average is negative 0.3, squared is 0.09. And finally, 0, no individuals in cell number 3, and so 0 minus 2.3, or equals negative 2.3 squared, 5.29. Okay, so now we're going to take the sum of all 3 of these values. Okay, together, it is 12.67. Okay. Now we have the numerator. The sum of all 3 of these is the numerator over the number of cells sampled, which is 3 minus 1, so 2. Okay, so 12.67 over 2 is 6.34. Right, so after all of that, we have calculated the variance. Right, how different is each cell from each other? Right, so 6.34. That's not done yet, right? The index of dispersion is the variance over the mean. And so, index of dispersion, variance over mean, 6.34 over the average, 2.3, is a total of 2.76. Remember that there are 3 different options. We have clumped, we have uniform, we have random. If the value, the index of dispersion, is greater than 1, we call the population clumped. And so that makes sense when we take a look at this. Here's a clump, and here's a clump, and here's a clump. So this is a clumped, very common distribution. So far, we have used the crude population density. Right? So just kind of taking an average of all of the spaces within the distribution of the organism. But this does not necessarily account for patchiness. That is, Some organisms can live here, such as the bobwhite quail, but they can't live over here. Okay, so bobwhite quail only lives in these hedgerows. And so if you were to take samples of random cells throughout the grid of this area, of this distribution, many of those cells would be here, right? So would be in these fields. In fact, most of them, statistically, would be in these fields. And so that crude population density is not actually telling us necessarily much about the organisms, right? Where they're living, how dense they are in the habitat that's actually suitable for them, okay? So the ecological density is a valuable metric for telling us how dense are those quail within the hedgerows themselves. Okay, similarly, if you looked at the entire distribution of this lovely little invertebrate here, you would think that they are everywhere, but in fact, they are only within, only on these poor tomato leaves. And so it tells us a lot more, in addition to crude density, it tells us a lot more about how many individuals these guys are interacting with on a daily basis. Right, and similarly, if we look at these yellow trees here, these cottonwood trees, many organisms live within these cottonwood trees and not out here in the surrounding arid environment. And so we can use ecological density as opposed to crude population density to understand more about what is going on within this microhabitat in the larger distribution for this organism. Measures of population structure. So far, we've been talking about simply numbers, like, is there a spider or is there not a spider? Is there a quail or is there not a quail? So that's just the true abundance, how many organisms there are, density, how many individuals there are in a space. But there's a lot more that we could talk about, of course. We can talk about the structure of the population in terms of age or age classes. Okay, so for example, if we look down here, we can see a human population structure in 3 different places. So Egypt, US, and Japan. Down here, kids under 5, and then older and older and older up to over 80. And so what we can see is the shape of these distributions changes depending on where you're looking. Okay. For example, in Egypt, we can see a positive growth rate. Right? We can see that the population is growing because most of the people are young and therefore coming up to reproductive age, or they are within the reproductive ages. Okay, so there are more people able to reproduce or soon able to reproduce compared to the older individuals that are no longer reproducing. And so this would imply that the population is likely to grow. In the US, this is about a 0 growth rate, right? You can see, about the same number of reproductively aged individuals compared to post-reproductively aged individuals. And if we look at Japan, we can see that there are actually more post-reproductive age than there are pre-reproductive age, right? So when this cohort enters into the reproductively aged, or into reproductive ages, they are going to replace these older people at a slower rate. Okay, so we can call these age pyramids, and they give us a little bit more information about what the population is probably able or probably will do in the future. In a future lesson, we are going to break this down a little bit more and talk about the actual growth rates. In some species, it makes more sense to talk about developmental stage as opposed to just pure ages. So for example, here, we see a tree population. We have many seedlings out in the open, and then very few, relatively few, that have actually been able to survive and live into adulthood. Okay, in the forest, you see more of an even distribution here, so relatively few seedlings compared to here, but many of, there are many more adults that are present. Okay, so this will give us a pretty good idea about the structure of the population and, in this case, the habitat as a whole. Okay. Another histogram here. Here, the age of this oak tree. In the forest, for the most part, the older trees are shading out the seedlings, the younger trees, and so you see no seedlings that are surviving, right? These are shade intolerant plants, but many more of these older aged trees. Okay. And so we can talk about this population structure in terms of the distribution through these age classes. Right? In general, in the forest, these taller trees inhibit the seedlings, and therefore this is a skewed distribution. And so mostly these older age trees, as opposed to here in the open site, where it would be skewed in the opposite direction. So that is all I have for you today. Thank you guys so much for watching, and I look forward to talking to you again. Thanks.